In this video I'm going to talk about multiple alleles and blood group inheritance. So this forms part of the genetic crosses series of videos, all for B3 for biology. Again, note that this is for triple biology, so for the triple scientists only. Now, up until now we've looked at individual alleles for two alleles for a particular gene. So say we've had eye colour, we've thought about two alleles, so a big B for brown and a little b for blue. Multiple alleles is when we're looking at more than two, the inheritance of characteristics which involve more than two alleles, more than two. And when we're thinking about blood group inheritance, we actually have an example of something called codominance. Up until now, we've had a recessive and a dominant allele. Dominant, recessive, if these are for eye colour. The capital is dominant over the lowercase, and so this would be brown-eyed, brown allele, blue allele, two different alleles for eye colour. Multiple alleles is where we're actually looking at more than just two, and a really good example is for blood group inheritance. It's actually quite complicated when you look at blood groups. We're going to do it on quite a basic level here. If you go on to A level, then you may do a bit more extension on this. So codominance is where, what if we had brown and a blue? This doesn't happen, but what if we had browny blue? A sort of middle ground somehow. And we get a little bit of this happening with our blood group inheritance. So that's codominance. And this means where two alleles are expressed or shown up in the phenotype. Remember that phenotype means the physical characteristics, the outcome of the genotype. A little bit of background then for blood types. Do you know your own blood type? I don't. Not many people seem to, really. Perhaps if you had a blood test you could ask. Blood types. There are four main blood groups that we have, and we're talking about in humans here. We have A, B, AB, and O. And the alleles that will determine which blood type you have. So the blood type is actually the phenotype, the outcome. And the alleles responsible, that could be in different combinations in different genotypes, the alleles that we've got, we write these alleles a bit like when we did the sex and characteristics. We have to write an X and a Y. We have to write a capital I. It's really important to try and make that look like a capital rather than just sort of a stick. And the I actually means immunoglobulin. You don't need to know this, but it's useful to know. Well, that should be an N. Globulin. And this just represents the structures on the outside of a red blood cell, which are called the antigens. So we can imagine that we've got our red blood cell there, biconcave disc, remember, no nucleus, and it's got little bits sticking out from it. Well, this is not a very good diagram, but it's got bits sticking out from it, little proteins. And it's what type of protein do they have on their surface? And that's why we write the little i's, just by convention. Maybe those little proteins, they might be of the A variety. They might be of the B variety. Or they might have none at all. So we're thinking about these little things that stick out on a red blood cell. So if we think about the alleles, well, we can have the little proteins that are A type, we can have the little proteins that are the B type, or we can have none at all, and we represent that with a 0 with an O. 3 different types of alleles. Look, it's multiple alleles because we've got more than 2 alleles, compared to our eye colour example where we only had 2 different types of alleles there. The dominance bit, so I've already said to you that we've got multiple alleles and it's an example of codominance. Well, here are multiple alleles. What about the codominant section? Let's have a think. If we think about the dominance, well, what we have is the A allele and the B allele are both codominant to each other. It means that neither is dominant over the other. They will both be expressed in the phenotype equally. And then we have the O allele. And this O allele is recessive to both A and B. Now, if we know that, we can try and do a genetic cross where we can try and predict what blood groups are likely from different combinations of people if we already know their blood group. So I'm just going to put this out of the way. I'll bring it back if we need to. So let's predict some blood groups. Remember that all of these genetic crosses that we're doing are just looking at probability. So with enough of these genetic crosses, this is what we would end up getting. But it doesn't mean to say that it's definite. It's a prediction. It's a probability. Let's have an example. We'll do a couple of examples. So I'm going to start off as usual with the parental phenotypes. Link's running out. That's disappointing. Okay, so I'm going to say somebody with blood group A, and I'm going to make them heterozygous. Oh, that's disappointing. That's my favourite pen. To try a barrow. Okay. Here we have. Sorry, I was going to say this was heterozygous. And somebody with blood group B, I'm going to make blood group B heterozygous as well. I can't write with this pen very well. I prefer my ink pen, but never mind. Genotypes. Okay, now this is a tricky bit. Again, hopefully in a question on an exam you'll be told a little bit more information. Now, if we look, if I want to have somebody who's got blood group A and they're heterozygous, if I look back, well, what genotype am I going to have? If these are the possible alleles, let's think about some of the genotypes that we can have. Well, I could be blood group A, and I could be homozygous. Or for blood group A, I could be heterozygous. And this is because, remember, that the O allele is recessive to A and B. If I want to look at blood group B, well, I can be homozygous, to the same, or I can be heterozygous. Remember, O is recessive to A and B. If I want to look at somebody who's blood group O, well, there's only one particular combination that we can have of those alleles, and that's this, and that's simply because the O is the recessive form. So in my question, I have said I want to have an heterozygous blood group A. Well, this will be this one, an A and an O. and a heterozygous B. There we go, that's homozygous. This one's heterozygous. I'm going to pop that one down. Alright. Right, so same as usual, same old gametes. Getting bored of writing all this, I'm sure, by now, but you'll be an absolute expert by the time you finish. Remember, we just separate these out because we have half coming from the mother and half coming from the father. And our gametes will contain half the amount of genetic information. That's why we're separating them out. Draw a little circle around them so we know for sure they are separate and we're not tempted to put both into our Punnett square. Notice I'm keeping my eyes as a nice capital with a top and a tail. So it's really obvious I'm doing it that immunoglobulin representation of our Punnett square. Draw my grid. Gametes, I'm going to label it. Just in case I've got extra points for labelling, I'll pop it down there. It's starting to get a bit scruffy now, but never mind. And I'm going to pop these two in the side. This one here. This one here. I'm going to pop these two in the top. IB, IO. Pop our little genotypes for our offspring in here. Again, we're in alphabetical order, I think. A and a B, an A and an O. Now we've got a B and an O, and we've got two O's there. Next stage, offspring genotypes. So write those out. This one, we've got an A and a B. This one, we've got an A and an O. This one, I've got a B and an O. This one, got two O's. Finally on here, offspring phenotypes. Now we may have to have a little think about our codominance here. Okay, so we need to remember that the A and the B alleles are codominant and they're both expressed in the phenotype, whereas it's our O that is recessive. I'm going to be thinking about that. I'm just going to pop that there. So our A and the B, they are codominant. So this means that the blood group would be AB. On this one, it was the O, which is recessive. So what would this blood group be if you have this genotype? What do you think? Yeah, it would just be A. And for this one, what do you think? Yeah, B. And finally, we have two O's. That must be the recessive phenotype O. So in this example, we have one of each. We have 25% AB, 25% A, 25% B, and 25% O. I'll do one more example on that, just so that you are confident with how to work these out. One more example. Let's do a parental phenotype. Let's say AB with an O. So again, that's our blood group, blood type. Well, what would be the genotype of those? Can you think what might be the genotype for somebody with the blood group AB? Have a think about the codominants. Yep, remember you draw the eye, the immunoglobulin, and an A and a B, they are codominant. And can you think what the alleles are for somebody with the blood group O? Yep, that's it. We've just got 2 O's there because it's the recessive condition. Let's do our gametes. Separate these out into our circles. We've only got one type here, so I'm only going to draw one circle because all these gametes would be the same. I could draw both out, it wouldn't make any difference, still get the same result, but I'm just going to draw one there. Punnett square. Draw my Punnett square. I'm going to label these gametes, gametes on the outside, genotypes on the inside, and look at these gametes in the side here, this one, IA, this one, IB. I only needed one of these, but it doesn't matter. Pop this one in here. Let's do our genotypes. So we've got an A and an O, and we've got a B and an O. Offspring genotypes. I'm going to write those out. You don't have to, we could just draw a little circle and label them, but just to show it nice and clearly, I'll write them out separately. This one, A and O. And we've also got one of these, a B and an O. Offspring phenotypes. Well, we've got an A and an O. Remember that A is dominant over O, so that would be blood group A. And B is dominant over O, and so we've got a blood group B. I'll write that there because the offspring phenotypes is what blood group is it. And we could do a ratio. We've got a 1:1 of A to B. Or we're going to have 50% likelihood of having blood group A and 50% likelihood of having blood group B. Hopefully that has demystified, oh sorry it's hanging off there, a little bit, the idea of multiple alleles. Just a quick refresher, multiple alleles, more than 2 alleles. And remember, when we do our normal genetic crosses, we've been just looking at 2 alleles for a particular gene, so more than 2 alleles that represent a particular gene. Blood group, we've got codominance, and that means that 2 alleles are expressed in the phenotype. Our codominant ones are A and B, and a recessive allele is O. We've got possible genotypes here, we've got homozygous and heterozygous, but we only have the homozygous possibility for O because this is a recessive, caused by the recessive allele, and you have to have 2 recessive alleles to have the phenotype of the blood group O. We can represent the inheritance of blood groups in exactly the same way that we've been doing all of these other genetic crosses, doing the phenotypes, genotypes, gametes, Punnett square, and then separating out the genotypes and the phenotypes in a ratio. In the next video, I will just do a few little, actually, I'll do it now, a few little final tips. Okay. No, I'm not going to. Oh. I'm going to. Okay. Final tips, right. Okay. Tips and tricks. What do we need to know? So all the things that I've been talking about across all of these videos is to write your gametes in a circle. Okay, something like this. If you're going to write the gamete, you do this. It's really important because it separates them out. Okay, what's my second tip? To label the Punnett square. I have seen questions where you've got marks for labelling it. People have missed marks if they haven't labelled it. So we also make sure that we know that we've got the gametes on the outside, and we've got the offspring genotypes on the inside. And by outside and inside, I mean this is the outside and this is the inside. What else has there been that's really useful? Oh, name the ratios. If you have a ratio of blue to brown eyes and you say the ratio is 3 to 1, fantastic. You get 0 points unless you also write down what the phenotype is. 3 brown to blue eyes. You have to clarify the ratios. Number 4, what might be my next tip? Well, a shortcut. If you get really good at these, then you'll recognise what the likely outcomes are of these monohybrid crosses. That's what we've been doing so far, the general ones like for eye colour. If you have a heterozygous parent and another heterozygous, these are just on our general normal ones that we started all the videos with, then you always get a 3 to 1 ratio of the dominant to the recessive allele. If you have a homozygous dominant and a homozygous recessive, you'll always have 100% dominant in your offspring. And that sort of makes sense because you're always going to have the dominant allele is always going to be expressed in the phenotype, whereas the recessive allele isn't. These ones you don't have to remember, but you'll sort of get used to them the more you use them, especially this one. I think this one saves you time, knowing that heterozygous times heterozygous is 3 to 1. That can really save you time, perhaps in a multiple choice, where you don't actually then have to go through and remember how to do the genetic cross. You see, oh, it's heterozygous times heterozygous. Right, 3 to 1, I know that ratio. If you had let's say a homozygous dominant and a heterozygous, we're talking about the genotypes of the parents here, then all of the offspring are going to be dominant, have that dominant characteristic showing in the phenotype, because you've got the dominance here, 2 alleles. And then a last sort of combination, if you have a homozygous recessive and a heterozygous, then you're always going to get 50-50. These ones, you're unlikely to memorise them. It doesn't matter because you can really easily work them out. I'd say try and memorise this one, saves you loads of time, and it also acts as a bit of a check that if you've ever done a genetic cross, which is heterozygous by heterozygous from monohybrid crosses, and you've got a 3 to 1 ratio, you know that you're absolutely on track. Another couple of things we've talked about on sex determination. Remember, you write the X and the Y first. Write those out first, because these are the chromosomes in the sperm and the egg. And if you write them out first, you're not going to then get confused when you're doing your genetic cross. If we have, we've talked about sex-linked conditions, and remember we've got our faulty allele. Where is the faulty allele? Where is it? On the X chromosome. And it means there's no portion, nothing on the Y. And remember, you have to write out your X and your XY first before you start adding any little letters on top of them. Write those out first. And finally, with the blood groups, we've just looked at those. This is an example of codominance, meaning that the A and the B together make AB blood group, and also multiple alleles, and that's where the A and the B alleles were codominant, were oh actually that's here, isn't it? They were codominant. But we also had multiple alleles. Remember, means more than 2 alleles. We have this extra little one, our O. And the tip with the blood groups and codominance and multiple alleles is you always write out your immunoglobulin to represent the little proteins that are on our red blood cell. You write those out first before you start adding your symbols for your alleles. There's been quite a lot there. I hope that this helps. Have a little practice, stop the video, have a think about how you'd write out the next bit, and hopefully this will help you with your revision and your understanding. Thank you very much.